This is the first screencast on the muscle physiology lectures. Usually we have two 1-hour lectures. What I'll do here is I'll break these two lectures down into a number of screencasts, probably about 3 screencasts for each. I think with these as well, we'll probably have some activities, but they're going to be integrated into the lectures, into the screencast, but we'll see how we go. So here are the lecture aims. What we're going to do essentially is talk about the 3 different types of contractile cells, or the 3 different types of muscle, namely skeletal, cardiac and smooth muscle. What we'll probably do for the first set of 3 screencasts is talk about skeletal muscle after a little bit of an introduction. Then we'll focus on the structure and function, particularly of skeletal muscles, as I said in the first 3 screencasts. We'll describe something called excitation contraction coupling. We'll do it first of all in some detail for the skeletal muscle, namely the neuromuscular junction. So we heard already about the excitation parts of excitation contraction coupling. This is the neuronal input, particularly these alpha motor neurons that go into the neuromuscular junction. We'll talk about the contraction, obviously the muscle part in these screencasts, and then we'll consider also excitation contraction coupling for cardiac and smooth muscle when we talk about those in the second set of screencasts. We'll also consider something called the sliding filament model. Sliding filament model we'll consider in some detail for skeletal muscle, and then we'll compare and contrast for cardiac and smooth muscle. Maybe just to say, if we do ask exam questions on this topic, remember that it's always good to integrate the topics. So the one reason we do nervous system and muscle system physiology almost together is because we want you to appreciate that these are very much linked, like lots of physiological systems. So in particular, when we're talking about excitation contraction coupling, remember not just to talk about neurons, not just to talk about muscle, but to integrate things if we ask you a longer type question on this content. So let's start off with a very brief introduction to the 3 different types of contractile muscle cells. As I said, skeletal, cardiac and smooth. So we're going to focus probably on the first 3 screencasts on skeletal muscle. We've already introduced the neuromuscular junction, so you've got an idea about what skeletal muscle is. It makes up around about 40% of the body mass, so it's very important. Obviously, it's responsible for movement, so it's linked to the somatic nervous system that controls our movement, which could be fine movement in terms of vocalization and moving fingers and operating tools, obviously very important for evolutionary development of humans as the predominant species on Earth. But also it's important because it provides protection and support, because it anchors the skeletal muscle to the skeleton. As I said, we'll talk about cardiac and smooth muscle in the second set of screencasts. Just to say here, cardiac muscle obviously is specialized for the heart, allows us to pump blood throughout the body and transport oxygen and nutrients. Smooth muscle is associated with our internal organs. So we've talked a little bit maybe about how the autonomic nervous system is important in regulating gastrointestinal tract function, but smooth muscle is obviously important in GI tract, equally important in things like the renal system, cardiovascular system and the respiratory system. So it's clearly integrally linked to some of the other topics that you'll cover throughout PM1A. I think this is, I've got a couple of diagrams here that I'll spend a little bit of time on, because I think if you know the if you be able to fill in all of these boxes in this type of table diagram, then you will essentially have a good summary, basic summary of what the 3 different types of muscle look like, what their characteristics are and how they're controlled. So the reason that skeletal muscle is sometimes said to be striated or striped can be seen here. So if we look at let's say a gross basic diagram of skeletal muscle, you can see it contains these stripes or striations. So it's striated, the stripes are visible in skeletal muscle as it is in the cardiac muscle. And actually one of the reasons, the main reasons that we call smooth muscle smooth is because it lacks these stripes. So that's a clear and obvious difference between skeletal, cardiac and smooth muscle, the stripes or striations. Perhaps the other, from a physiological point of view, the main physiological difference is obviously we know that skeletal muscle is under voluntary control because it's neurogenic. It's got this neural input, whereas cardiac muscle and smooth muscle, probably as we said in the previous set of screencasts, are myogenic. So they're not controlled voluntarily. They're under involuntary control. We're not thinking about controlling our heartbeat. We're not thinking about the functions that smooth muscle control, control of the airways, control of the guts, control of the renal system, all of those things we saw briefly outlined on the previous slide. Again, there are some morphological differences, if you like. Morphology pertains to shape, essentially. So skeletal muscle cells are elongated. These are often branched. You can see the branched here, and these form a kind of spindle. If you don't know what a spindle is this kind of shape here. Something that you wrap thread round is a spindle, tapers towards the end. That's what spindle means. And again, the number of nuclei and location is different for each of the different types. Multiple nuclei tend to be located peripherally, obviously, because we're concerned with muscles on the outside of the skeleton. Single or double nuclei, which tend to be located centrally. Single nuclei located centrally. Sorry if that was a bit confusing here. This is more of the location of, this is pertaining to the location of the nuclei on the periphery of the muscle organisation, which we'll talk about over the next few slides anyway. Again, this table looks a little bit complicated. What we'll probably do is I'll put it up here, but maybe come back to it at the end and indicate some of the important proteins, maybe some of the important aspects that we want you to know about the differences between skeletal, cardiac and smooth muscle. So again, this might look a little bit daunting at the moment. We'll talk about skeletal muscle and hopefully you'll see where proteins like troponin fit in, why we're mentioning these proteins down here, the dihydropyridine receptor, the ryanodine receptor, why they're important in skeletal muscle, but maybe not so important in cardiac and smooth muscle, how the control is achieved. We've kind of talked about this already. Alpha motor neurons, whereas these are under control of the autonomic nervous system. Something about the control. Again, as we talk about excitation, contraction coupling, maybe we'll see why some of these fibers are important throughout the systems, what the difference is in calcium binding proteins, maybe between skeletal muscle and the other types of muscle. All of this hopefully will become clear as we go through the next few screencasts. Probably if we were in the face-to-face lecture at the moment, I would ask you to have a look at this, chat to your neighbour and see if you can identify the three different main types of cells. As this is a screencast, you can pause the screencast, see if you can identify each type of muscle. So which one is cardiac, which one is skeletal, which one is smooth. Have a little think about that. I'll just pause for a little bit and then I'll give you the answer. So if you could identify A as skeletal, B as smooth, and C as cardiac, then you're off to a good start. Again, you know, have a look back at the previous slides if you couldn't get that right. So in addition to, so we're going to focus on skeletal muscle for the next couple of screencasts. We'll talk about a little bit about organisation and excitation, contraction, coupling at the neuromuscular junction. So skeletal muscle, as we said, as well as being involved obviously very important in the motor system, in movement. Also controls things like respiration, because the diaphragm, this big muscle that controls breathing in and out, is skeletal muscle, and also has important homeostatic functions. So we've heard about homeostasis in other lectures, this balance that the body needs to maintain. The muscle system, in particular skeletal muscle, is also important, has important homeostatic functions. Bone is able to store calcium, other ions. Skeletal muscles can also store potassium. Also important, obviously, in things like metabolism and temperature regulation. Temperature regulation, maybe the most obvious thing there, is shivering. So if you get your skeletal muscles contracting, those are the muscles that are involved in the shiver response, which can regulate temperature. Yeah, so what we'll do over the next couple of screencasts is talk about the tissue organization, particularly for the different proteins that are involved in skeletal muscle contraction. And again, we'll spend a little bit more time on skeletal muscle versus cardiac and smooth muscle. And then maybe we'll compare and contrast the proteins that are involved in skeletal versus the other two. We'll talk about excitation contraction coupling, and you know, as we said before, excitation pertains to the nervous control, which we've covered previously. That's why that's grayed out. Contraction is the muscle contraction, and as I said, we'll concentrate on the sliding filament model. Let's just conclude this screencast just by giving you some characteristics of the two main types of skeletal muscle fibers. So you can see in overview here we have red fibers and white fibers. The reason they're called red is probably, if you focus in on here, you can see that these are these muscle fibers, which my pointer is hovering over, are slightly more red color than this whiter color that you see in white muscle fibers. Both surrounded by things like blood vessels and adipocytes. Essentially, the main difference between, the red and white fibers depend on the type of activity that you want to undergo. So we use red fibers for certain types of activity, and white fibers for certain other types of activities, which will hopefully the next few slides will lead you to. I don't want to tell you what they are now, because hopefully you'll be able to deduce them from some of the properties. So energy can be made quickly without using oxygen, using glycolysis. Again, these are things that you'll cover in probably other parts of the lectures. For longer periods, we use different processes, things like oxidative phosphorylation. And again, we decide physiologically which set of muscles to use, red or white, depending on the type of activity that we want to engage in. So if we want fast dynamic activity, or maybe slower activity, we use different types of red versus white fibers. Red or white with distinct properties. So let's consider red to start off with. Red muscle fibers, shown here, found in what we call postural muscles, so things to do with maintaining our posture, surrounded by adipocytes. Diameter is about half the size of white fibers. We haven't got a scale bar on here, but just to tell you that they're slightly smaller than white muscle fibers. The reason they're red is because they're very high in myoglobin. So myoglobin is a protein that's found most highly in red muscle fibers, surrounded by capillaries, lots and lots of mitochondria, the energy source, but low glycogen. So red muscles are efficient. They produce sustained amounts of energy, and they can maintain contractions for long periods, hence why they're called postural muscles. The contraction rate is slow. This is because they use oxidative phosphorylation for energy production. We'll see this contrasts with the white muscle fibers as we go to the next slide. So for this reason, they're sometimes called slow or oxidative muscle. So if we contrast that with the white muscle fibers, they've got this white muscle appearance. They're found in fast muscle, and that's basically because they use a different type of energy production, which we'll see down here. They're white or pale due to the lower myoglobin content, so they haven't got this red appearance, surrounded by fewer capillaries and relatively few mitochondria, but they're very high in glycogen. So they rely obviously on glycolysis to produce energy very quickly. So they're said to be fast muscles. If they're fast, it means that they can tire, so they can be subject to fatigue. They're fatigable. So they're called fast or glycolytic muscle fibers. So yeah, here's maybe an activity that I would probably ask you to do if we were in the classroom. Here's, this is a much more, let's say, stylized diagram of the difference between red and white fibers. Here we can see in these slow twitch muscle fibers, predominantly they are red rather than white. If we move then through to maybe a mixed population, then a predominantly white population, what I'd ask you to think about now is, in different types of activity, which type of muscle are we going to use? So if you want to, let's say, have a fast dynamic sprinter response, which type of muscle fiber would you use in comparison to, let's say, different activities, walking, jogging, in comparison to sprinting? So I'll have you just have a little think about that. You can pause the screencast if you like, and then I'll give you the answer. So essentially what you should have recognized is that red fibers, let's say in terms of long distance rather than kind of jogging, I guess. I think the important thing is, if you want a fast dynamic response, you should identify that these fast twitch white muscle fibers are more important because they produce energy very quickly. But they're also fatigable. You can't keep sprinting for a long time. That's why predominantly if you're jogging or more long distance running, you're going to be using your red muscle fibers more than your white muscle fibers. What we should also think about maybe is that that instead of thinking of clear differences that we maybe intimated between our red muscle fibers and our white muscle fibers, is that there is a continuum. So in terms of slow twitch muscles, we have we can also see in textbook they're called type 1 muscle fibers, through to type 2, which are fast twitch. So type 2A and type 2B, there are slight differences. So if we think about these as our red fibers, their resistance to fatigue, to tiredness, whereas our white muscle fibers are fatigable, as we said before. Slow twitch use oxidative phosphorylation, whereas fast twitch use glycolytic. So white uses glycolysis, red uses oxidative phosphorylation. Because of this, or this is dictated by the availability of mitochondria, high in red, low or fewer in white. Glycogen is low in red muscle fibers, but high in white muscle fibers. And so these type 2 fibers are somewhat intermediate. So maybe it's best to think, if we go back, this is our maybe intermediate situation here. So we've got a clear distinction between red and white. These intermediate fibers have intermediate properties. So whilst their resistance to fatigue, they're relatively high in mitochondria. That's why they're also known as red fibers. They use oxidative phosphorylation, but glycogen is somewhat intermediate, so they can contribute to the functions that were associated with white fibers, which is why they're predominantly classified as fast twitch. So I hope that's clear. Okay, that's quite a long introduction. We're coming up to 20 minutes, so I'll stop there. The next two screencasts will be slightly shorter and consider the organisation of skeletal muscle and excitation contraction coupling in particular.